So I'm going to talk today about virtual microscopy project that we are working on at the Academy of Natural Sciences of Philadelphia, which is, by the way, one of the oldest natural history museums in America. So here you can see the long list of my co-authors. These are mostly Drexel undergraduate students who were all contributors to that project. So what it is about? So first of all, I want to say a few words about our collection, just in case somebody is not aware how important it is and how wonderful that collection is. So Diatom Herbarium at the Academy of Natural Sciences of Philadelphia, which now belongs to Drexel University, is one of the oldest and largest collections of diatoms in the world, if not the largest. It dates back to mid-19th century. And here you can see faces of our founding fathers and one mother. So Christian Faberger, Charles Boyer, Frank Keeley, Charles Reimer, Ruth Patrick, and you can see the years when they were active in the collection. You can see that pretty much they devoted their largest part of their lives, decades of their lives, to establishment and growth of this collection. And because of their and their successors' vision and efforts, we now have this amazing resource. The collection has several hundred thousands of diatom samples and slides, including about 5,000 type slides. An example of such a type slide is shown here. This happened to be a holotype slide of two species described by Dr. Patrick. And because of these amazing holdings, of course, this collection is a valuable resource for studies in diatom taxonomy and systematics, in water quality assessment studies, and in paleoecology. You, of course, are aware that most of biological research collections these days are engaged in digitization of their specimens, because these digital specimens are very convenient data source. They not only allow the remote access to specimens, but they also promote collaboration between scientists, and also promote automating of data collection, data gathering. So usually digitization of specimens like herbarium sheets or preserved animal specimens means taking 2- or 3-dimensional images of these specimens, sharing them online. And diatom collections also been involved in these efforts. For example, we digitize more than 400 type specimens more than 10 years ago and posted them online. But what happens with diatom specimens, especially bigger diatoms, the thicker specimens, it's often insufficient to have just one image. So, for example, you see here Auliscus schultze, the diatom described by Boyer, and it's a big diatom. So you have to take images at several focal planes, and we resorted to just posting them next to each other. On top of that, diatom slides contain valuable information at the level of assemblage. And that's a very important resource for studying diatom occurrence, patterns of abundance, diatom abundance, and distribution. So it's important to digitize not just individual specimens, but whole slides. Unfortunately, now, because of the advent of this new technology, whole slide imaging and virtual microscopy, we can do that. This technology was initially developed for pathology applications. The field is known as digital pathology, but also sometimes it's referred to as telecytology or telepathology, because you can share the images of slides in electronic format in very fast way. So that allows collaboration between doctors and pretty much allows for faster medical diagnosis, which you can understand it's very important, right? So if you're facing some disease, dramatic disease, you want to have your diagnosis as soon as possible. So the workflow for whole imaging, slide imaging, involves preparing glass slides, feeding them into automatic slide scanner, and then taking these multiple images at many fields of view across the slides at different focal planes, then stitching these individual images called tiles together in one three-dimensional image of the slide, storing, sharing these images online, and then analyzing them. And of course, you can imagine these days it's AI is involved into analysis workflows for these types of imagery. We can adopt that technology, but there is a difference. If you're working in biomedical situation in the pathology lab, you're making slides and you can make them in totally uniform way, so it's the same size and shape and content pretty much. So you can use these automatic scanners that do not have any ocular view. So it's not like a real, it's a microscope, but without ocular view. In museum settings, we are digitizing old slides made by different people in different ways, and they're all different, not uniform. So we cannot use these automatic scanners, which are actually getting relatively cheap. So we have to use microscope, like standard microscope-based equipment. So that's the difference. Actually, the first diatom researcher who started whole slide imaging of diatom slides was Bengt Betseri, who worked at that time at Husted's collection in Bremerhaven. And together with Mike Kloster, they initially applied whole slide imaging technique to study of Fragilariopsis populations from Southern Ocean. So this is the figure from their 2017 paper, which shows like a standard scan. And they used research quality slides. So they're good quality slides. You can see diatoms more or less clean, not obstructed by any debris, not overlapping with each other. So they're nicely distributed over the slides. So like you can see that the species of interest, the taxa of interest, which happen to be Fragilariopsis here, they're outlined by blue. So they actually selected these images on the slide and analyzed them. So the workflow that Bank and Michael developed for their study included first scanning slides at relatively low resolution, then manually or semi-automatically selecting specimens of interest, then rescanning these parts of the slides at higher resolution, and then using algorithms. First, they used Sherpa, then some more sophisticated algorithms for extracting features. And then you could be morphometric features or anything else. And then you can use this data, that information, for any to address any research questions that you want. So they, for example, this is from their 2018 paper on Fragilariopsis. They used this morphometric data for taxonomy of Fragilariopsis. So here you have like groups of specimens grouped by species into 3 species. Later, they continued applying this technique for answering other research questions, for education, for constructing training data sets for automatic diatom identification, but not really for like digitizing collections. So important feature of the workflows that Michael and Bank developed is stacking of individual images of diatoms taken at different focal planes into a single image, extended focus specimen or image. And then you can use that image for extracting features, and that works pretty well for the research purposes. You can then use this single flat image for displaying on different image viewing platforms. In particular, they like using the Beagle platform. Um, because it's a single layer image, you don't need any very sophisticated image viewing platforms. And as I said, that's having that perfect image, like it's meets the research requirements. But on the other hand, it requires good quality slides, unobstructed specimens, not broken, not having debris on them. But our tasks for our projects are kind of different. First, our project is about digitizing museum specimens. So we need to make our images absolutely publicly accessible. So if you want to see these images on Beagle, for example, you need to have an account, you have to have access to particular sets of images. We wanted totally free public access. So you don't, like in our case, you won't need any special permission or even setting up any accounts or anything like that. Second, we wanted to provide the user experience as close to examining real microscope slides as possible without manipulating images in any way and leaving this opportunity to users. So we wanted to preserve these different focal planes, not using any stacking. So when I started investigating the field, what's happening in biological research collections, if anyone is using this technology, I started thinking about that in around 2017 and 18. At that time, what I found was just only two collections, biological research collections in the United States actually started using whole slide imaging. And both collections happen to be invertebrate zoology collections, one at Field Museum in Chicago, and the second at Yale Peabody Museum. So that project is led by their collection manager, Eric Lazovasim. So at that time, myself and Alison Minerovic, who was our collection manager at that time, we actually went to Yale, we visited Eric, and we were absolutely impressed by their progress of their team, and by their setup. So this slide actually shows a specimen of a bryozoan that from their collection of digitized slides. And you can see that they actually didn't need very sophisticated viewing platform. They used pretty simple code, but it works perfectly for their specimens. So this particular display allows you to move through multiple focal planes. But then you can have another specimen, and it's slightly different display. So here you can zoom in and out of that image, and you can also move across the slide. But in that case, you won't be able to move through focal planes. So it's either one or another. Eric was extremely helpful in guiding us and helping us to figure out how to proceed with this project and what to do. And because we were so impressed by their setup, we ended up purchasing the same equipment, hardware provided by this company based in UK called Infor in Images. So this is how our setup looks like. So this is Olympus microscope, just standard microscope, but it's equipped by motorized stage. And then you have a computer with software, which is actually running this motorized stage and taking images. Our stage has only 4 slots for the slides, and it perfectly works for us because we still have to examine each slide before we decide what area on that slide we're scanning, if that slide is amenable to scanning at all, because some of our slides are so sparse that doesn't make sense actually to digitizing them, because you're just waiting wasting file size for digitizing empty spaces. So we skip digitizing very sparse slides. And then we are using this surveyor software which runs the stage. That software was produced by the company called Objective Imaging. And so that's how the scan actually proceeds. That's what you see on the screen when you're doing the scan. So this is just one field of view. And here, this monitor is just showing you like how individual fields of view are imaged. So on each stop, the software runs the stage through multiple focal planes and collects multiple imaging. So that machine has been running pretty much nonstop for the last couple of years. And this is another scan, like where you collect more focal planes. So that would take maybe half an hour, like that's a longer scan. So it's anywhere between maybe like 10 minutes and half an hour for scanning individual slide. However, the most technically challenging part of that project was preparing these files, these image files, for displaying them, for sharing them online. So we have this amazing team of Drexel undergraduate students who were actually writing the code for displaying our images. And first task was to figure out what image viewing platform to use. As I said, we wanted to preserve that capability of moving through focal planes at the same time moving across the slide, zooming in and out, all these three capabilities together. So we had finally, like the students identified that platform. It happened to be this Lumenera platform, open microscopy environment, obviously developed first for displaying digital pathology slides, but turned out it works perfectly for our purposes. And then these two students, Hang Duong and Rohan Chandrakatni, they wrote the whole code for stitching the individual tiles, these individual images together for making these three-dimensional images. And then they also had to write a file conversion scripts to make these files compatible with the Mera format. And then Hang also wrote a code and configuring that virtual microscopy website for our purposes. So that's how it looks like from our side, like this Almera website where our images are displayed. So on the left, you can see the list of the individual projects, how many scans are in each of these projects. On the right, you can have, you see metadata for a particular scan in the slide. So that one is for icon, which is selected in the middle of the screen. So that one particular scan shown here. But then Han also developed a public view of the same Almera website, which is more convenient for external users to use because it's not overburdened by this detailed information. And it also has this search capability. So you can, in that window where you see search, you can type like any term which is included in metadata for slides and imagery, and you can pretty much go select the scans from that view. So for example, here I searched by Vermont, and then I got a long list of scans from that state. And on the left, you will have icons for each scan. And on the right, it's metadata for that particular slide. So it's whatever was in our database, like state, county, coordinates, and sometimes like type of the sample, depth down the core. That's what we had in that case. Then you can click on the icon for that scan, and you can see the next screen where you can slide. So the slider on the left allows you to move through focal planes, and then you can zoom in and out, and you can move across the slide. And here is an example of how you're zooming through one particular scan. So this one happens to be a sample from sediment core from New Jersey Lake. So you can keep zooming on it, and look at these images. So what we achieved so far? So we scanned around 5,500 slides so far. And these scans mostly represent slides from projects run by EPA and USGS. And you can see here, I listed the names. So USGS National Water Quality Assessment Program, EPA National River and Stream Surveys, lake samples, mostly core sediment samples from EPA IMAP project, New Jersey DEP coring project, EPA National Lake Assessment Project. This is only from the eastern part of the country. We chose to focus first on these big federal and state projects simply because they have abundant environmental data associated with diatom samples, and therefore they present a valuable resource for all kinds of studies in diatom ecology and taxonomy. We mostly did our scanning using brightfield illumination and mostly at 400x magnification. And of course, we didn't scan the whole slide. So when I say whole slide imagery, doesn't mean like really the whole cover slip, like, so our scanned area, standard slide scanned area is 19 square millimeters. Smaller subset of slides were done at 100x or 1000x, in that case with oil, but still using bright field illumination. We can do DIC illumination too. It just, the whole setup requires much more work and time consuming, but we plan to do that later, like playing more with setup and when we are focusing on the types in the future. So while working on that project, we, of course, came across multiple technical challenges, and it was expected because this is pretty much a pilot project. So the first challenge, and it's a very serious challenge, it's a huge file size. So when you do the standard scan at 400x with 40x objective, the size of the file that scan is around 30 gigabytes. It's huge. So we have to compress these files when we do the file conversion, making this our scans compatible with Amera format. But we experience some loss of quality. I should say that when our students were writing the code, they experimented quite a lot, trying to to minimize that loss of quality. So it's not a huge loss of quality, but still, it would be great to some days, to store the original scans. But our servers are just have limited capacity, so we cannot do that. Then we have kind of cumbersome file handling process, and it's partly because we use this custom assembled equipment and also because of our IT infrastructure limitations. This challenge is not as fundamental as the file size, and with more IT resources, it could be circumvented, but it just requires more resources in IT department. So frankly, the summary of our efforts, I would say that we showed that this digitization of diatom collection using this whole slide imaging is possible. But there are still a lot of work to improve the infrastructure, and we need to address these long-term maintenance challenges. Finding the storage more sustainable technical solution, that would be important. So what we're going to do in the future, the project, actually the NSF funded project just ended. So now we have some time to think what to do in the future. But immediate needs, we want to make our scans more easily discoverable, sharing the links to the images on different biodiversity portals, and in our own collection database website, which is a new one. It's now, the collection database is available on Symbiota portal, And it's our collection manager, Laura Aycock, who is responsible actually for developing that site. Laura already inserted links to these virtual slides to several thousands of the images. So you can see in the bottom of this slide that I'm showing right now, like that one particular record for one slide. And you have this little tab called associations. And that's where the link to the image is sitting. So the virtual microscopy image is sitting. But we also want to share on the other biodiversity portals like IDIC Bio, like GBIF and others, and also on Dona. We, of course, will continue searching for sustainable technical solution for this virtual microscopy. We want to scan more type slides and various historical collections that we have here at the Academy. And also, we want to use scans for public outreach and educational purposes. And some of our virtual microscopy slides have been already used for exhibits at the Academy. Like, for example, this image of arranged slide, a beautiful arranged slide. This is virtual microscopy slide, and it was already used in an exhibit. And some of our scans, because they're three-dimensional, if you think about this imagery, it's three-dimensional imagery, they were already used for making holographic images of diatoms and also used in one of the exhibits at the Academy. And also, like the next task is to make educational products. For example, this was our attempt to create kind of a lesson based on Mark Edlund's study of sediment cores in Lake St. Croix in Minnesota, where Mark published several papers, and Mark and his collaborators, I would say, like just looking at how diatom assemblages changed since the time of European settlement. So two undergrad students from Drexel who were involved in our project, Sylvia Lepik and Hannah Domingos, they scanned slides that Mark gave us representing these sediment cores. They also studied papers and they wrote a narrative, like linking images to interpretation, what you can say about environment looking at these sediment cores. And this is also like the images are all available online. So, for example, if you just look at the list of these samples, you see the like what's available. You can click on the icon. Again, you see the view of the slide, so you can change magnification, like move the slider across focal planes. And then on the right, you see like the like you can see annotations. So like we annotated some of these slides. So for example, like if you want to see how Stauroserella leptostauron looks like, if you want to show to your students, you can just direct them to that particular place on the slide. You can zoom on that image and you can see that Stauroserella leptostauron. You can let your students to play with this image, maybe try to identify diatoms. You can also snap an image. I actually forgot to mention that earlier. Amera allows you to snap images of what you're seeing on your screen. And you can do whatever you want with these images, analyze them in all kinds of ways, like smooshing them in one single file, whatever you want to do here. So here, like I annotated several taxa, like showing examples of the taxa. So that's what we are doing here. Finally, I would like to thank anyone who supported that project, starting from, of course, with NSF that funded the whole project. And two people whose help I especially appreciate are Eric Lazowassim from Yale Peabody Museum and Mike Broderick from Info and Images. Without their advice, I don't know how I would be able to deal with this project. INS IT department is responsible for maintaining our servers. And here you can see like the beautiful faces of our contributors, mostly Drexel undergraduate students who scan the slides, develop the code, and make all this possible. So thank you for your attention. Now I think I can take any questions. Maybe I should. Am I still sharing?
No, it's not sharing, but that's great. Thank you so much, Marina. And if anyone would like to ask a question, you can raise your real or virtual hand or put a question into the chat. And maybe I could start. Oh, we have one from Julianne. So, do you have a student who would like to develop an AI program to scan the historical periphyton slides for Didymo? That's Didymosphenia.
No, I don't have a student who is, because I'm not that technical, right? So I see my role is actually like making this available. Anyone who wants to participate, we will be happy to collaborate. But I think it should be the next generation of, either diatom researchers, but it's mostly like coding work, right? So you need a programmer to do that, working together with diatom people. So anyone who wants to use this, collaborate with us, you're very welcome. I'm not doing that myself, and I don't have any student who is involved currently into that.
Marina, I'd love to talk to you more about the flow cam technologies for recognizing and clipping images. That software, if anyone's familiar with the flow cam, really the main thing that they do is recognize shapes and clip them and organize them. And I'm really curious about if some of the existing code can be developed for what you've done.
I think that's exactly what should happen, and actually what my cluster developed, I think it's their algorithms are doing the same thing. They're just kind of recognizing the tags of interest by outline, I believe, or maybe by some other features, and then clipping them.
Mm-hmm. Maria says, thank you for such specific update on these new technologies, methodologies. And Sylvia asks about taking a snapshot of a specimen. When you take a snapshot, does the image include a scale bar?
Yes, yes, it does. Actually, it would be nice if you guys, whoever is listening here, if you could try, like use this, go to images and play with them. Like everything is available online. It's Amera is open. Okay, I can. I don't know if I can. Maybe I should put the link in the chat.
Marina, you and Mark developed a teaching model based on the slides from the Wisconsin lakes. Can you describe what's involved in that exercise?
In the lesson, so it's pretty much like a narrative, like that's what Sylvia and Hannah wrote, and I edited a little bit, just like describing what Mark did, like the summary of the research, and then just screenshots like showing like where you can go, to particular slides, and what you can do, like how you can move through the slide, how you can focus on particular specimens, and depending on who is actually practicing, who is taking that lesson. If it's somebody who had no clue about diatoms, it's probably best to direct these people to these annotated specimens, so they can go there and say, oh, that's how Stauroserella leptostauron looks like. And then you could go to Donna and read about this species, right? So it could be like training like that. If it's more experienced person, that person can just use these slides for trying to identify diatoms, right? So you don't need any microscope. You can just zoom into the image, and if you notice, the quality is pretty good. although it's not oil objective, it's just 40x objective. But look at that Storozerella leptostaurans or whatever it's. So it's not like that lesson is not targeting a particular audience, like narrowly defined audience. I wanted to keep it kind of like open and like anyone can use it for like adjust it, like adapt to their own level.
But it is set up that somebody could go through and essentially do their own analysis of that core.
Absolutely. Yeah. That's possible too. You can do the count, just moving like you would be counting the atoms.
Yeah, that's fantastic.
And we can compare the counts, whatever.
Anybody else? Tom says, thank you, Marina, and your team for making these slides available through the web. Are type slides available in the same diatom.symbiota portal?
So Symbiota is the collection database, and we just started it recently. Laura just moved our database into Symbiota, and it requires still quite a lot of work, but a lot of our types are in that, they were in that old database since the time like Charles Raymer and Ed Terrio put them in that old database. So they are there. The scans we didn't scan most of the types, like using whole slide imagery, right? The flat images of the type specimens, they used to be available in the old interface, which is not working right now. So it's now up to Laura to kind of like make the links, to these images, but many of them are available.
And can you describe that again? You showed the types. If somebody clicked a link, they would get an image of the type. So sometimes they might be whole slide scans, and sometimes they might be what you did long ago, or all of the.
Right, exactly. Yeah. Sometimes it could be like one or another or both. Yeah.
Is that linked through the new taxon file or?
No. New taxon file doesn't have anything. It's a separate resource. It's different from collection database. This is about collection, digitizing collection, keeping records of the collection. Mm-hmm.
Marina, I had a question. So if somebody would like to send you a slide for taking imaging, like in processing, is it possible to do that? And how would that person like approach to do that?
You mean the whole slide imaging?
Most likely, but like you said, it's not the whole slide, but it's like 18 something square meter, right?
It's not like we have like a lot of resources for imaging like big sets of slides. But if it's like a few, 1-2 slides of which are particularly interesting for some reason, like maybe type slide, yeah, we can do the scanning.
Okay.
We cannot add like thousands of new scans now because very soon we will reach the capacity of our servers.
Mm-hmm.
So if you want to scan like thousands and thousands of new slides, we will have to find more storage space somewhere. And that's an open question how we do that. I don't know. I don't have a solution for that.
Marie, do you have an idea of the cost of that? Of the server space?
When we were buying our servers, we spent like more than $20,000 on them. There are 2 servers involved here. But I think the cost of the storage goes down. And there are some other technical solutions. You can use the cloud, like what, I don't know. And it's also the matter of scale. If it's like a huge resource available somewhere, through like some government source, I don't know, like if it would be cheaper, per scan. People are
Yeah.
scanning, like the tissue scans are also like immensely huge. Like people are scanning, like, for example, thin sections of like the brain, like that level of like cells, neurons, like, and they're huge. I know Google was saving, like, I don't know, storing these biomedical images, but this is about human health, and it's still like challenging.
Anna has a question. Can it be a monetized service that I guess the Academy scan for someone who sent the slides and pay a fee for it, and then you don't store them at transfer to the owner of the files?
Yeah, theoretically, yeah, that would make sense. It's just storing in different places. It's technically kind of difficult because like the pipeline, like streaming the images to the imaging platform is just written for that particular server, right? So somebody needs to create another pipeline, pretty much.
But you already have, what was the total? Thousands of slides that you've scanned.
Yeah, it's almost 6,000. Yeah.
Almost 6,000 slides are already available. That's incredible.
Yeah.
Tom adds maybe historical blooms and future blooms. He's thinking about the first pseudonychia bloom that was recorded to result in widespread injury in Canada in 1987.
Interesting. Yeah, that's an interesting idea. Yeah. There are a lot of interesting things that could be, like digitized, like it's the technical part which is like really difficult, like finding that solution, and that requires collaboration with engineers pretty much.
I like the idea of just people exploring what you've already posted and trying to use it and see how it works.
Yeah, that's what we want to get, like more feedback, like try to use it actually, like for something.
Yeah, I think the idea of making it a class exercise too. Thinking about a way that it could be sort of distributed. You have a class and one student in the class analyzes the slide for one taxon. They get to know that one, and another student does the next. I can think of a few ways that would be a sort of a interesting way to start to distribute the work, just like people do that with cataloging stars.
Yeah. Yeah.
Well, anybody else have comments for Marina or our Diatom Web Academy sessions?
I just posted the link here to that public site, just in case people want to copy it.
Great. I also put into the links, there's a project page that you put together, Marina, and I think we have all of those links on that page. I'll check it again. If we don't,
actually we need to update because actually we just changed the servers, and that's another technical challenge. The servers keep, like, the IT department keeps updating the servers, so like in the middle of the project they were required, because of cybersecurity challenges, they were required to change the servers, and it took forever. So that's why it's kind of slow, like, so we just changed the links. So now that's the correct link now. So we'll have to update the links. Yeah.